Hello everyone, this is Dhanashri from the Dr. Omics Research Lab. In this topic, we are going to discuss about the quality control using the FastQC tool. As we have downloaded the raw data using the SRA database, so this is what a FastQ files we are having, which is of paired and data, so forward and reverse file we are having. And now for this data, we want to check the quality that whether this data is having a good quality or not. Maybe this data is having any outside sequence, any adapter, any primer or not. So that's why we are going to check the quality for it using the FastQC tool. So we just have to open the terminal from the folder where we are having our FastQ file, and we need to install the FastQC using the sudo apt-get install FastQC by the repository. Once the installation is done, to check whether the tool is installed or not, you can just type the fastqc and enter. So here it will open the manual for it. Fastqc space hyphen hyphen help. So once you'll enter, it was going to open the manual. It means the fastqc is installed. And how to use it? So basically, you just have to call the tool fastqc, and then you need to mention your samples like your forward sample or and reverse sample. Otherwise, you can simply use the command fastqc star.fastq. Star means what? All. So it is going to check the quality for all the files which is having the .fastq extension. And now why fastqc? Because it is providing us a very simple way to do some quality control checks on our raw data. With the help of graphical representation only, we can understand whether our data is having a good quality or not. Because obviously in a FASTQ files, we are having so many reads, so it's not possible to check a quality for each and every read, and that's why we are going to check a quality graphically. So it's very efficient tool to check the quality for the raw data. And now you just have to run this command. You can use any one of it. So it is going to check the quality for the forward file and the reverse file as well inside the raw data folder. So once it is done, after that here it is going to generate two files for each sample. Out of that, one is a HTML file and another is a zip file. But HTML file is giving you the graphical representation, and the zip file, it will give you the summary for the graphical representation. So if I will just open this HTML file in a browser, so here it is going to show you the quality. This is a quality for the forward sample, and in the left side they have given the summary, or you can say these are the parameters on the basis of which we are checking the quality of our raw data. So this parameter here, if you will see, it is showing you green sign. Some parameter you will get as a you are getting here as a red cross mark. Maybe some parameter you will get as a yellow exclamation mark. So it is indicating the quality. Means what? If your parameter is having a green sign, it will give you means like for that parameter your data is having a good quality. If a parameter you are having is with a yellow exclamation mark, it means for that parameter your data is having a moderate quality or average quality. And if your parameter is having a red cross mark, it means for that parameter your data is having a poor quality. It is indicating the error sign. But to decide the quality, we are not focusing on all of this parameter. We are focusing on per base sequence quality. This is our first main important parameter. Our second main important parameter is the overrepresented sequence, and the third one is the adapter content. So these are the three main adapters. Other than this, if any parameter is having a red cross mark, you don't need to be worried about it. So if you will see here the per base sequence quality. So basically in the first parameter, they have given you the basic statistics, which is a file name, what is the file type, then what encoding they used, how many total number of sequences they are having. Total number of sequences means what? A total number of reads. Then sequence flagged as a poor quality. So here there is no sequence which is flagged as a poor quality. Sequence length, it's 301. Means what? It's a read length. Most of the reads from our sample is having this length and the percentage GC content. So this is the basic statistics which is giving you the basic information. But now let's go for the first parameter, the first main important parameter, which is a per base sequence quality. So this per base sequence quality, it will tell you the quality as per the base. You can see x-axis, it is showing you the position in the read, that is base pairs, and y-axis, it is showing you the Fred quality score. At this basis, they are represented by the whisker plots or a box plots, and these are the coral tiles of the box plots. So now, as this is what a forward file we are having, because this is the paired end data, so you need to be check the reverse file as well to decide the quality. So I'm just opening this reverse file as well here. And you can see in the reverse file also, the bases they have represented with the whisker plots and a box plots. So box plots, actually it is giving you the range, up to what range the value is going or the intensity is going. And this y-axis, if you will observe that it is divided into three colors like green, yellow and red. So this is a quality. Like if your base is falling under the green area, it is indicating the good quality. If your base is falling under the yellow area, it is indicating the moderate quality, average quality. And if your base is falling under the red area, here it is indicating the poor quality. And if you will see all the bases in this, they are falling under the red area, nearly a 3 Fred quality score in between 2 to 4. And that's why this parameter is showing you the red cross mark. And this is also, if you will see, this coral tiles is also falling under the red area, but here it is not showing any whisker plot, so it is just showing you the green sign, that's why. But still for the reverse file, if you will see, bases are not having a good quality, so we can't say this parameter is good quality. Now our second main important parameter here is the overrepresented sequence. Overrepresented sequence means what? Like if any sequence is overly representing in your data, so that's what you will be able to see here. The table is given here. So this is the sequence which is overly representing, how many times it is overly representing, what is the percentage and the possible source. Now, if you are getting such kind of overrepresented sequence, it's totally up to you that you need to trim it or not. But if in a possible source it is showing you no hit, at that time you don't need to trim that sequence because these are the sequences from our data only. These are not outside sequences, and that's why you don't need to trim that. But in a possible source, if you are getting the uh, words like a adapters or a primers, so at that time you need to surely trim out that sequence because those are the outside sequences which you can't keep in your data. But right now, if you will see in this table, we don't have any uh, possible source with the adapter or primer. So here also in the reverse file also, you need to check that because this is the paired data. So we can say that this is a this is fine. Like you don't need to do the trimming over here. But the last one here, the third main important parameter, which is the adapter content. So if any universal adapter is present in your data, at that time you will get the graph. Red color graph, which is representing the Illumina universal adapter. If you are getting the blue color graph, this is the Illumina small RNA 3' adapter. So if you are getting such kind of adapter, you need to do the trimming over here. And from this, we can conclude that or we can understand that actually our data is not having a good quality. We need to perform some trimming over here. So that trimming part we'll discuss in a next part or in a next video. So if you have any doubt, please let us know. Till then, bye. Have a great day.